This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 682, recorded on November 12th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hello there, Vincent. Special day today, and it's 75 degrees and partly cloudy here in Austin, Texas.
Well, we have a really special episode for you today. We are bringing back a guest who was previously on TWiV 444, which we recorded. Rich and I recorded with our guest at the American Society for Microbiology meeting. Was it in Chicago, Rich?
No. It was in New Orleans.
New Orleans.
I remember because I flew there. That was a special trip. I just flew in for the day to interview Kate.
So we had interviewed Kate Rubins, and she had finished her previous mission. Well, what, 8 months previous to our TWiV then, right?
Yes, that's right.
So her last mission ended 4 years ago. That was 48/49. And I remember when we talked with her, Rich, she said, I don't know if I'll go up again. There are a lot of astronauts.
That's right. And now Kate gets a second mission. So for those, as background for those who may not have listened to that episode, and I recommend that people go listen to that episode. Kate was trained as a molecular biologist, but from childhood had always aspired to be an astronaut, but kind of, life got in the way and science got in the way. So she kind of forgot about that and went through the whole shtick, PhD and postdocs and stuff, and wound up at the Whitehead doing virology and in fact working on pox viruses. And then the opportunity came along to apply for astronaut training, and she applied and was accepted and did a mission, as we said, on the space station. It was about 5 months long. 4 years ago, and we interviewed her after she came back, and she's been very gracious in communicating with us, and she's just fascinating to listen to. I love this space stuff.
And she was the keynote speaker at the ASM meeting that year, and she was as the keynote. She was interviewed by Ed Yong, the science writer.
Oh, I didn't see the keynote, so I didn't know that. That's great.
Yeah, that was. They both sat in chairs up front, and she showed some video clips of her mission, and then he asked her questions. It was quite nice. So here we go. We're about to interview Kate. She's up on the space station, and there's a couple second delay, 2 to 3 second delay in the audio. So it's not our usual conversation. Rich and I have a series of questions. We're going to alternate. But this is a first for TWiV. We're really happy that NASA lets us do this, that Kate lets us do it. So let's check it out.
Station, this is Houston. Are you ready for the event?
Houston Station, I am ready for the event.
This Week in Virology podcast, this is Mission Control Houston. Please call station for a voice check.
Station, this is Vincent Racaniello with This Week in Virology podcast. How do you hear me?
Hey, Vincent, this is Space Station. I've got you loud and clear.
Our guest today was previously on TWiV, only now she is much farther away from the International Space Station, Expedition 64, Flight Engineer Kate Rubins. Welcome back to TWiV, Kate.
Thank you very much, Vincent. And I have to say, it is always perfect weather up here. It is 22 degrees, and that never changes.
That's just great to hear. Let's dive right in because we don't have a lot of time. My first question for you, what special expertise do you bring to ISS science, and what influence did you or your expertise have on the structure of this mission and its science?
Thanks. That's actually a great question. So NASA selects people from a variety of backgrounds, fighter pilots, engineers. I happen to be a molecular biologist. I used to be a virologist. I hope I still am. And so I bring a lot of biology expertise to the station. But as I'm up here doing experiments, I'm learning material science and physics as well. So I think general science experience and then more specifically biology and molecular biology.
Kate, great to see you. In our previous interview, you said that as a kid, you wanted to be an astronaut, a biologist, and a geologist simultaneously. Now we see that one of the experiments on this mission is bioasteroids, studying the interaction of microbes with rocks. So you're in fact doing all three simultaneously. So could you tell us about the bioasteroid experiments and their purpose and what do you expect to learn from them?
Yeah, it's really fun to be up here. You get to be all kinds of scientists at once. The bioasteroid is actually looking specifically to see if there's microbial interactions to do mining, so extraction of rare earth elements via bacteria and fungi. And we're looking at this in microgravity. NASA's interested in this, obviously, for future exploration. We want to see if we can do what's called in situ resource utilization. So when we go somewhere, we're obviously going to have a lot of lunar or planetary material available, but we need other elements. And so this experiment sets us up to try to do biological mining for future exploration.
Can you tell us how cell culture, tissue, and organoid culture has advanced since your last visit?
That's a great question. Cell culture up here is not as easy as it would seem because fluidics are so different. So fluids float. Obviously, if you're going to try to set up your normal 6-well cell culture plates, take the lid off and the fluid's going to wick up like capillary action. Some of it's going to float out of the plate. So the way you do cell culture on Earth just doesn't work up here. When I was here in 2016, we were working just on developing long-term culture methods, seeing if we could do it, seeing if we could grow cardiovascular cells in space. And we proved that we really could do culture for up to 30 days. I didn't contaminate the cell cultures. This was great news. It's also not so easy to do sterile technique up here. We can't have ethanol. So that 70% ethanol spray bottle you're used to spritzing your hood down with is not allowed. It's flammable. So we have to wipe everything down with BZK wipes. Sterile technique can be both harder and easier in space. It's easier because once you've got everything wiped down, you can just let the object float. I'll show you with the microphone. But it can be harder because everything's floating. Obviously, it could have some contact after you've gotten it wiped down. So we really just established that we could do cell culture up here. Since then, it's advanced significantly. So we can send cells now frozen and bring them out of the -80 and start a culture line, which greatly helps out if we don't have to ship up fresh cells and launch them from the Earth. It really expands the amount of cell types we can send up here. And we can do both 2D standard traditional cell culture and 3-dimensional culture up here. We can grow organoids, and we're going to be doing a lot more of that on this mission.
So, Kate, I'm wondering if cells grown on the space station display any structural or biochemical abnormalities, and if so, what those are, and how do they inform your perspective on spaceflight?
Yeah, it's one of the things that we're really interested in. Structurally, there definitely are, I wouldn't say abnormalities, but there's differences. So anything in a fluid environment here tends to just become very spherical. So that this happens with cells also. They become much more spherical. And so this, we think, obviously, is due to changes in cytoskeleton. We also have this issue where it's not a drawback necessarily. Cells that would normally kind of settle down to the bottom of the plate remain in suspension. And so you can have these suspensions of cell culture without having to do a rotating vessel. So we kind of try to suspend cells on Earth by doing these rotating vessels, putting those cells in free fall, essentially. But when we do that, we have a lot of fluid shear forces. And so on the space station, this whole thing is in free fall. And so if we bring up some cell culture, it's in free fall without that rotating wall vessel. So we don't have any fluid shear forces on those cells. There are definitely changes in gene expression. whether that's due to some of these structural differences, differences with adhesion on the plate, differences due to microgravity, possibly differences due to the low level of radiation up here. We don't know, and so there's a lot of things that we're investigating now about gene expression changes on board in cells.
You were the first to sequence DNA in space. How has sequencing technology advanced in 4 years? And can you now routinely process samples, like from a microbiome experiment on board, all the way to obtaining a sequence?
Yeah, that was really one of the drivers of the sequencing experiment is microbiome. I mean, if you got a sequencer, why not sequence the microbiome up here? It's great. So we do microbiome samples as our standard part of our environmental monitoring. We check the station periodically, and we can monitor for any runaway microbial contamination or fungal contamination. But right now, we have to send those samples all the way down to Earth, and then they do standard laboratory techniques. We can only send them back to Earth when we have a vehicle that's undocking and landing, which is not that often, every few months. So we wanted to have figure out what the bug is right now, kind of a sequencing technique. And that's really advanced since I've been up here. So what we were able to do is a culture all the way to sequence. And then we're also able to do a culture independent. So just take a swab, process the sample on board, go all the way through sequencing. Not only have we done DNA sequencing, we've now done RNA sequencing. And there was a very cool student project that actually looked at DNA repair. So they had yeast, induced a break, and then repaired it via CRISPR and followed that whole process with sequencing. So we have a lot more planned, but they've really done a lot in the last 4 years.
So for the listeners, I want to make sure everybody understands that the microbiome is the full complement of microbes at any given site. For example, the human gut or skin or any surface such as your dinner table. And I'm wondering, Kate, what's the status of the microbiome experiments on the space station?
Yeah, that's a great question. We have done microbiome experiments. We've looked at both surfaces of the space station as well as humans. And those experiments are ongoing. So we're starting to do that with every crew member now. The initial experiments were very episodic. So we were taking a few samples and we were looking at that through the mission duration, but it wasn't a huge number of samples. That paper has been published. I'm sure we can get you guys a link to that for the show notes. And the interesting thing, it's, fairly predictable, but I think we were interested in seeing how the microbiome evolved over time. It seemed to become less diverse, and the crew members' microbiomes seemed to start resembling each other more. But those were with a few number of samples per increment. So what we have planned in the future, and one of the things that I'll be doing when I'm up here, is a really dense sampling. And so we want to do very dense spatial sampling of the station. We have different modules. I'm talking to you from the Japanese module right now, but we have many different modules on the station. They have slightly different surfaces, and so we want to see how things vary on the different surfaces. And then, of course, we're very interested in the human microbiome. So our question is, how stable is that? If we get a really dense time course, can we see small fluctuations? And we're particularly focusing on time periods of change. So new crew arrival. Does my microbiome change when I get my 4 new friends who are hopefully coming up here on Sunday? And how does that affect microbial diversity?
So would you know yet if there is a ISS microbiome signature?
There are differences in your ISS microbiome versus your Earth microbiome. I think we'll be able to call it a signature when we get more dense time points and sampling, but certainly there are changes from our pre-flight and our post-flight.
So us being virologists, does the microbiome measurements that you're doing include the virome? And if so, what do you observe in terms of viruses?
Of course, that's my favorite thing to talk about as well, and it does include the virome. We did look at virome samples. This is a big shotgun approach. The virus our samples look to be about 1% or less of our total samples. And so one of the next things that we're doing is specific targeted amplification. So we know from previous papers that weren't just whole genome sequencing, but very specific PCR type papers, that we do have viral reactivation in astronauts in long duration spaceflight. So we have VZV reactivation. We've had causes shingles. This is the chickenpox virus, and we've had cases of shingles actually up here in spaceflight. We've seen EBV shedding, and so we have a number of viruses that we're interested in sequencing, but with a little bit of amplification to see if we can pull those out of the background. And then I think we want to do some more. We've been doing a lot of skin type sampling. It's really good to do more saliva sampling to look at most of that viral shedding. And so actually every morning this week, I have woken up and I've taken a saliva sample as part of one of our studies that's going to look at the virome over time.
So switching subjects a bit, how many plant habitats are there and what are their purposes?
We have several plant habitats. So veggie is a plant habitat where we can grow plants, we can observe them. They use these, obviously dirt would float here, so you can't just plant on the ground. So they use these special, they call them plant pillows. And it's kind of a cloth mesh fiber that covers the dirt, and then we can grow the plants out of that. And we have a very specialized facility. It's called Advanced Plant Habitat. I've been working a lot on that last week and this week. That's more of an incubator where we've got a sealed atmosphere in it. It's like an incubator door, and we can control the atmosphere. We can control CO2. We can control humidity. It's got special lighting. And so we just planted last week some radishes. It's a great experiment. They're similar to Arabidopsis. They grow very quickly. They produce a lot of material, and so they're interesting from both a plant biology perspective as well as a possible crew food source for the future.
Yeah, so you anticipated my question about growing radishes in space. That's pretty interesting. The material on your mission specifically describes what I guess is a new habitat, Plant Habitat 2. Can you tell me what that is and how it differs from other habitats already?
So that's got a lot finer control of these environmental conditions. And so obviously plants like high CO2. People don't like high CO2, and we struggle with that a lot in our spacecraft atmosphere. We want to bring the CO2 as low as possible for the human beings, but we want to keep it higher for the plants. And so this is a special incubator with a seal in the door, and we can precisely control the CO2 conditions inside that habitat and keep that atmosphere separate from the rest of the space station. Also humidity, and then we can fine-tune the light source, light cycles, and wavelengths in this habitat.
Do plants grow normally in space? And if so, if not, how do they differ, and how might that affect space travel? Yeah,
it was previously thought that root growth was really strongly influenced by gravity, and this was hypothesized by people on the ground that thought that gravity was one of the factors that would make the roots go down and the plants go up. And some of our early experiments up here showed that actually gravity is not that strong of an indicator. It's not a very big influence on the root growth. So plants grow fairly normally up here. They're most sensitive to things like their atmospheric conditions and then, of course, their light source. And so we think, the plants that I planted last week are looking good. We got a chance to pull them out. There's 3 seedlings per well, and there's 20 wells. So I did some thinning the other day to just pick the best plant and let that keep growing for the experimental conditions. But they are looking good. They're looking as good as anything I plant in my garden on Earth.
So I'm probably accessing the book The Martian for this, but I'm wondering if you can recycle human waste on plants. And conversely, if the plants could potentially be used to process human waste purposely, so you'd have your own more self-contained ecosystem.
Yeah, that's a great question. You think of the same thing up here as well. So right now we can't use the plants to process human waste. We're concerned about, obviously, microbial contamination, and we don't have a really great way to sterilize human waste up here, solid waste. So we're not using that for the plant matter, but we can use the plants potentially in the future to help us with CO2. You would need a huge amount of plants. So you would need something like a plant wall. NASA's definitely thinking about that kind of thing for exploration. I'm guessing we're going to try some larger plant habitats here at some point. So I think the most promising thing is probably looking at our atmospheric cycle with the plants and having them help absorb some of the CO2. In terms of the solid waste up here, we recycle everything up here, right? So our water recycling is really incredible. It's a 90% closed loop. So we take yesterday's coffee, make it today's coffee, make it tomorrow's coffee, and we lose very, very little water. We reclaim all the condensate we've got a condensate bus that cycles through the whole station, and we reclaim that, and we can put that, once it's clean, back into the potable water bus. Solid waste we can't do such a good job with right now. So the current methodology is to collect it, put it in a vehicle that's returning, not landing, but burns up in the Earth's atmosphere. And there are small microparticles of our solid matter that just burn up, and that's how it goes away. But that's not great recycling. So every time I think about this, I think, it would be really good if we could sterilize it and use it as 3D printer material. I hope somebody is working on that. I have not seen any experiments as of yet.
Kate, our time is up. Thanks so much for joining us. Have a great mission, and we'll see you on the ground.
Absolutely. And just wanted to let you guys know, we've got TWiV up here on the space station, and you all keep me company on the weekends as I'm floating around getting stuff done.
Station, this is Houston ACR. That concludes our event. Thank you.
Thank you to all participants from This Week in Virology podcast. Station, we are now resuming operational audio communications.
It was a blast to see her up there, wasn't it?
Oh, yeah, it's great. Just wonderful.
I think it's cool that she started with the weather, right?
Yes, perfect. I wanted to ask her about the weather, but I didn't want to suck up that much time. But
yeah, she. That's cool that they listened to TWiV, right? Great.
yeah, actually, before I forget, I want to extend my gratitude not only to Kate, but to all the people at NASA. There were a lot of people who went out of their way. I mean, it's their job, but still, to make this happen. And we had some sort of ideas about how we wanted to do this that weren't in their normal thing. And they didn't say, no, you can't do that. They said, okay, we'll figure it out. So they were terrific. And I really want to thank them.
So when we were talking with Kate, the space station was around the very southern tip of South America, right?
Yep. And I've wondered, and I still haven't gotten the answer to that. I haven't really asked whether the timing of this, they gave us exactly 20 minutes. And certainly she was essentially as overhead as she was going to get. But I assume that it's easier to do when there's a more or less a direct line to her rather than being on the other side of the planet. But I don't know that, but it's interesting.
Yeah, and if you saw me giving hand signs to Rich, I had off to my side here, Amy Rosenfeld on the phone with one of the people, I guess in Houston, and she was telling her the time, 10 minutes, 5 minutes, 1 minute left. And so I was giving Rich signals so that we would not go over, because they'll just cut us off, right? Yeah. They're pretty strict about time, which you can understand. So anyway, thanks to Kate Rubins, thanks to NASA, thanks to our support people, and thanks to you, Rich. You're the one who organized this.
Hey,
great.
I love it. This is great. And I hope this is not the last time we talk to Kate. It'd be nice to have a casual conversation with her again. And once again, I in preparation for this, I listened to that old episode that we did with her, and it has a lot of detail, a lot about her experiences and stuff. And I highly recommend that.
Yeah, one of the things I remember she talked about is how they learned to deal with water in space. So when you pipette into a tube, right, it turns out if you put the pipette on the inside of the tube, the water flows right down the side of the tube. It doesn't just flow everywhere. And, if you've seen videos of astronauts up on the space station, they can take a wet washcloth and squeeze it, and the water just wraps around their hand because of the surface tension. It's amazing. And so she talked a lot about pipetting and centrifugation and so forth on TWiV 444. You should check it out.
There are a few other tidbits that I really liked. One was she talked about how every action, of course, has an equal and opposite reaction. And so she discovered that when she tried to pipette in space, the action of pipetting moved her backwards. So she had to hook her feet. And the other thing that one of many things that I really liked was readjusting to being back on Earth. And she would drop her phone because after 5 months in space, she was used to just, letting go of her phone and it would be there, or whatever tool. And back on Earth, she would just let go and, oh no, it drops to the ground. So great stuff.
So she's going to be up there for 6 months. It's a long time.
Long time. Oh, and for those who are interested in this, if you poke around in NASA, and I have an appropriate app for this, you can find various apps that'll tell you when you can see the space station pass over. And it's usually at dawn or dusk, so that it's dark enough to see it and close enough to the sun so that you can see the sun reflected off the station. And it takes about 6 minutes to streak across the sky. It's got to be high enough. But I recommend doing it, and wave to Kate.
It's a big object, this ISS. What did you say? 3 football fields?
Something like that.
Wow. And it's surrounded by solar panels, of course.
She's doing a couple of spacewalks on this trip too. So we'll
neat. Cool.
Remember she called that type 2 fun?
Yep. Well, that's cool.
Afterwards, it's fun, but in the process, it's a hassle.
Anyway,
I'll keep an eye on her and we'll keep you updated. I get updates as to what she's doing.
Do they release videos of these spacewalks? Do you know?
I think sometimes I'll keep an eye on it and we'll see what we can do.
All right, that was TWiV 682. Did I get the number right? 682, yeah. If you like what we do, well, first of all, that was TWiV 682. You can find the show notes at microbe.tv/TWiV. Send your questions and comments to twiv@microbe.tv. And if you like what we do, we'd love your financial support. You can go over to microbe.tv/contribute. Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Kind of a nice break from politics and pandemics, right?
And it's a good reminder that I was thinking about this morning. This is real science, okay? This is where we're curious. And so we're orbiting the Earth and doing experiments and stuff. And it's also a reminder, I mean, this is a cooperative effort. It's the whole, Kate went up in a Soyuz spacecraft. Okay, and the European Space Agency is involved. On Sunday or Saturday, they're going to launch Crew Dragon that includes a Japanese astronaut. So this is what can happen when people cooperate with each other. It's a beautiful, beautiful thing.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. I should also say, actually, that NASA recorded parts of it, and I'll probably use their audio and video in the final product as well. So an unusual twist. I get a little bit of help. Thanks, NASA. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.